Okay, so thanks for coming for the last day. So the plan today, so we have two lectures. So in the first lecture, it will be mostly slides, and it should be pretty visual, so slides are better. I will tell you, and today I will tell you mostly about my own work on evolution of networks. So the first part will be mostly on what I would call Evo-Devo. Which is networks related to development and how you can model development and use the approach which I'm going to describe, that is evolution in silico. And in the second part of the talk, I will tell you about some more recent work I've been doing on immunology, immunorecognition, using the same kind of idea. And it will be more on the blackboard because you can do simple but cute calculations. So you should have received the notes for all my lectures. So there are some slides, there are also some handwritten notes. I apologize for the handwriting. I was not planning to send you the notes. It was mostly for my own use, but we thought it would be useful for you nevertheless to have some written record of what I said. Okay, so I'm basically going to start with where we stopped yesterday, and with a question which you can ask, which is are all motifs born equal? So yesterday already we started discussing this. In the context of synthetic biology, we saw that some people have been designing small motifs to perform dynamical functions such as bistability or oscillations, and the motifs they use are not necessarily the ones that are present in actual networks, biological networks. And also the other thing we saw that there are, if you have even with the small motifs with few nodes, you can have many possible dynamical behaviors. And so bottom line is, we don't quite know a priori, which motifs do we expect to see in networks. So we can analyze them statistically, but we cannot really know a priori. And it would be good to have a way to, thinking in terms of motifs, finding a way to say something a priori on the motifs you expect to find in networks or something like this. And so with this question, this question has actually, it has been one of the central question of my work since my PhD. Okay, yeah. Before I go this, it's really related to this question I asked early in my very was that my first lecture? I don't remember. Maybe my first lecture, which is this idea that I was describing the Gould experiments, thought experiment, that if you take the evolutionary history, go back to Cambrian, and imagine you will be able to play it back again, how the animals would look like the animals you actually observed. You could ask the exact same question at the network level. Imagine I take the network that we have today, we go back to Cambrian, we let them evolve again. Would we see the same network structure? You could really ask the same question. And so, as I say in my first lecture, you need to have a formalism to do that, to do this experiment. Of course, you cannot do these experiments in real life, but if you have a formalism to study this problem, you could actually try to study this problem in a more theoretical way. And this is what I've been trying to do for more than almost essentially almost 15 years now, starting during my PhD with Vincent Hakim, where we essentially played this game of evolving networks, but in the computer. So we simulated exactly Gould's experiments. It's not exactly Gould experiments because we did not go back in time and went forward. We were mostly interested in forward evolution. Although later today I will show you an experiment of going back and going forward. So we've also done that in a more precise context, but for now we're just interested in the forward evolution. And so the idea we had is that these networks evolved, so we could just simulate evolution of networks, and what is really important that you want the network to change. It's not, okay, sorry. I'm getting excited. Okay, so we want network to change. We want to simulate an evolutionary process with networks. So you start, so the idea is that you start with a very simple network. You put it in a computer, and you try to simulate an evolutionary process in the computer. And I'm going to detail exactly what I mean by this. And the hope is at the end, you will end up with a very big networks, similar to what you see in nature. And the important thing, again, is that we want to create new networks. It's really the idea of macro evolution that I described 2 days ago. We want to see how new motifs, new structures emerge in these networks that we evolve. And okay, so that's the idea. And so let me now describe exactly how you do that. So first, in terms of formalism, mathematical formalism we've been using, it's exactly the same kind of formalism that I used on the blackboard the last couple of days. So we simulate networks with simple mass action laws. So now you should see what I mean. This is exactly what I use for transcriptional networks. And so, for instance, and we use standard biochemistry to describe interaction. So this equation here is describing essentially the biochemistry of this protein B in this network. So what does this network mean? It means that you have an activator, a repressor A repressing the transcription of B, and then A and B form a dimer. So that's basically a mixed feedback loop, as I described yesterday. And so if you have such a mixed feedback loop, you can write a differential equation for B that would look like this. And now if you go term by term, we can associate these terms to actual interactions. So for instance, this term here, this is our Hill repression function. It's written a slightly different way, but this is a repression of transcription of B by A. Okay, so we've seen that earlier this week. Now these terms here are just mass action law terms for interaction, protein-protein interaction. So A plus B gives AB. So this term here is basically consumption of B by A, and so this is proportional to A and proportional to an association rate. And this term here is basically the backward. So AB can dissociate into A plus B. I haven't indicated it on this scheme, but this is a dissociation, backward dissociation of the complex AB. And then, as usual, I have my degradation of B. So this is a kind of formalism I'm using, and so. I'm going to evolve this network. So that means that I can associate. So it means I'm going to change the networks, and I can add or remove interactions in the network. And anytime I add an interaction, I'm going to add terms in the equation. So you could also think about it in terms of, optimization of coupled ODEs, where you add terms in the equations to get a behavior you're interested in. Okay, so that's the formalism. Okay, so now how do I perform? I told you we want to evolve networks. And so to do that in the computer, we basically implement a Darwinian selection scheme. And so we start with a population of different networks. So these networks are going to be independent. It's like different individuals in the population, very much like what you've seen in this population genetics classes that you had this week. So you have a population of different networks. And so to each network, you have, each network have different dynamics. So for instance, here there is a feed-forward loop network. Here is just a mixed feedback loop. They have different dynamics, so have different equations. And so then what you can do with the computer, you have your equations, you can simply integrate the equations. And so let's assume we get the behavior of these networks, we get something like this. So it's a completely fictitious example. I don't claim this as the actual behavior has anything to do with the network topology I drew on the slide before, but imagine you have a behavior like that. So these networks have different behaviors. This corresponds to the phenotype of the networks. It's like simple Darwinian selection. You have different genotypes that gets you different phenotypes. And so now how is evolution working? Well, you want to select for something. You want to make a selective pressure. The goal here is to try to see if when you select for specific functions, specific dynamics, you see some networks emerging. And so, for instance, imagine I want to put a selective pressure to select for oscillators. So, visually, you would say maybe these two guys are better oscillators than those two guys. So you need to have a metric to quantify that. This metric is difficult to define because if you want to have an oscillator, if you want to quantify the oscillating characteristic of this, well, it's not obvious what to do if you don't have already an oscillator existing. Clearly these things don't oscillate, but they might have ways to tell, well, this is closer to an oscillator than these guys. So I come back to that later. The fitness, the score function that we define to perform your selection is actually quite important, and obviously it's important because it's going to define what is the selective pressure you put on this population. But it's also important because if you choose a bad way to quantify this, you will be in trouble, and you might get evolution stuck. Okay, so but imagine I have this. Imagine I have a scoring function. I have something that tells me that these two guys are better oscillators than those two guys. So then what you do very elitist Darwinian selection. You simply eliminate the guys or the networks that you don't like because they don't behave properly. And then what you do, you duplicate the networks that are behaving properly. And once you've done the duplicate, you mutate them. And so mutation can be, for instance, you change a parameter in the equations, or it could also be you add nodes or interactions. So that's how you do this evolutionary computation. Okay. So just to summarize, the goal here is to see how networks can evolve and how many motifs can emerge in complicated networks. And so to do that, you basically simulate in the computer an evolutionary process. So I've just detailed a give you a very bird's eye overview of the way you do that. You do a selection process on a population of networks. The networks are defined by ordinary differential equation. You do random mutations, you do the selection based on a scoring function or a fitness function, and the hope is that after many, many generations of this process, you will end up with a complicated network, and this complicated network, maybe it's going to be one of the motifs we found, or some variation of the motifs we found, and then we might be able to go back to biology and say, well, in this context, I expect to have this kind of motif, because my simulation suggests that we should get that. Okay, so if it's a bit abstract now, I hope that later on it will become more clear, because I will show you actual examples of how this evolutionary process works on real life problems. Any question on this stage? Is it okay? Okay, fine. Okay, sorry. Okay, so this is a small technicality. I'm going to skip that, but this is something that is, very briefly, when you do optimization, any kind of optimization, you need to choose properly the function you want to optimize. And so this is a problem that we have here in evolution. If I want to select an oscillator, again, how do I choose a good scoring function that will quantify the oscillating characteristic of the networks? It's not a trivial problem. And so you need to think about that. I will give you some more detailed explanations later on. Okay, so so we have developed a software to do that. So this is my kind of advertisement for my software. So this is called PhiEvo. This is basically the brand new version of this algorithm that I've been developing for almost 15 years now. It's in Python 3. You have Jupyter notebooks, stuff like that. If you're into these things, it's quite good, I think. Quite good. No, it's useful. It's very helpful to have things like that. It's on GitHub, so you can find the links to the code at this address. And so I even made a tutorial video to explain you how to use this evolutionary code if you're interested in this. So this code is basically implementing the algorithm I have just told you. And I went pretty quickly, but there are many technical issues, like how you encode equations, how you add links, how you mutate the things. That essentially we're doing this for you in this code, so you don't need to care. You just need to. What is important is that to the problem you're interested in, what you want to evolve in the computer. Okay, so now what I do is that I give you examples of biological problems where we use this approach of evolving network in the computer and seeing what kind of networks are coming and what kinds of motifs are appearing. And so I give you now biological examples, and we'll discuss them, and you'll see how I think we got interesting results. So in the first part of my lectures, so my first lecture, I'm going to focus on development. And so, and more precisely, on something quite interesting that you see here, which is, so it's very visible on the whale here. So if you look at the spine of the whale, clearly you have many metameric units that are repeated, and these are the vertebrae. And so this is some common to all, all vertebrates. They always have this vertebrae repeating here. Of course, the body plan also is derived actually from the first layer, the structures, and so you see it common between these animals. And so also one thing that is very striking, which is indicated here. So this in scale, you have almost like 2 orders of magnitude, maybe 3, between these different animals. So even though there are, massive difference in size, the way their body is organized is the same. So that's a problem for biology. How do you do that? And how do you, how account for scaling? And how are you making sure that, the vertebrae are all the correct size? How do you don't want to make a vertebrae of, well, your hummingbird, obviously. And how do you, how are you building this body plan, essentially? Okay, and so we're going to focus on this segmentation part, which is how you build essentially the vertebrae and how you build them identically. Later on, they will be specialized, but in the beginning, in the embryos, they're all the same essentially. And one important thing is that this process of formation of the vertebrae, it's called segmentation. And this is as old as animals. It's really like, remember my slides about Gould and going back to Cambrian. At Cambrian, the animals were already segmented. So this is something almost as old as life, as metazoan life, essentially, the segmentation. So it's a fundamental process that you would like to understand. And so I'm going to skip that. So this is our possible ancestor. It's still along the same line. So this is an Ascidian. This is called Picaya. This is possibly our common ancestor. And so you see already in this animal that is, more than 500 million years old, you already see, these little segments. And you also see that these days in modern Ascidians. And so you see that you form the segment. So it's something very old that appeared very early, a fundamental process of life. And if you compare it to frog tadpole, so you don't quite see the segments, but you can guess the segments here. So anyway, it's an old process, and I got interested into this process. So the classical paradigm for segmentation, this is something you've seen last week, it's fly. And so with John Reinitz, you saw this, I think, no, it's not from Reinitz. It could be from Reinitz's lab, I don't remember. I should have put some credit, I apologize. But you saw this classical paradigm of fly segmentation or fly patterning. So as you saw, you start with bicoid and other maternal genes. Then the maternal genes activate gap genes. Okay, so here, for instance, you see hunchback. This is hunchback. This is a gradient of bicoid. This blue thing is like bicoid getting above hunchback. And then when there's no hunchback, you have nothing here. And then, so you lay the gap genes. So I think you focus mostly on, with John Reinitz, you probably focus mostly on gap genes. But then later on, down the cascade, you have other genes like Eve, FUTS, engrade wingless, and so these genes essentially lay a periodic pattern, so like stripes, and these stripes are going to define future segments. Okay, so this is the classical paradigm for segmentation. This is a fly, that you make stripes, and in the end, due to gap genes and maternal genes upstream the cascade. So this has been a very well-studied problem. These people have worked on that for 30 years. What really was actually the identification of the gap genes was one of the key steps in the study of this problem, because you realize that you could lose gap genes. So did John Reinitz tell you why they call gap genes? It's because if you remove this gene, you basically have a gap in the body plan. It's like you remove a gene and suddenly so for instance, if you remove hunchback, basically you lose the thorax of the fly. You just go directly from head to essentially abdomen. So that's why it's called gap genes. So it was really it was really striking when people discovered that you could remove part of the body plan by just mutating one gene. And so this is why it has been so well studied and so important in the history of developmental biology to focus on this segmentation paradigm in fly. The problem is that fly is a monster. There are very, very few animals actually developing like fly with this gap genes and segmentation gene and stuff like that. I mean, and I'm going okay, I should take this back. I should not say there are very few animals developing with gap genes and segmentation genes. What is true is, as illustrated, it's not like you have an egg of fixed size and then you have the maternal genes defining the gap gene and essentially you lay the pattern. The way most animals develop is like this. So this is a chicken embryo. It's courtesy of Olivier Pourquier, with a very famous biologist. So before we start again the movie, this is the future head of the embryo. This is the tail of the embryo. And you'll see, you should focus on this region, and you see that this region is going to extend. And as it extends, you're going to form segments. Now I move. So you see this is called a tail bud. The tail bud extends. And as you extend, you form vertebrae in the upper part of the embryo. So that's very different from fly, where the fly is essentially a fixed egg, and then you make the pattern, you make all the stripes, and at the just before gastrulation, essentially you already have defined all your segments. Here, in most animals, what you do is that you define segments as you grow. It's a much more dynamical process. That's the thing you should bear in mind. It's a dynamical process. So this is all vertebrates, develop like this. All goes through this extension of tail bud formation of vertebrae. It's very, very well conserved. The precursor of segments in vertebrae are called somites. In case I use these words later on, I probably use this word because this is the term. So somites are, and they are the future vertebrae. So somites, vertebrae, it's not exactly the same thing, but if I tell somites, you should think precursors of vertebrae. Okay, and so that's true for the vertebrates, but actually most arthropods, they develop like this as well. So even fly within arthropods is an exception. If you look at tribolium, for instance, tribolium, it's exactly the same kind of process where you have an embryonic elongation, and as the embryos elongate, you form segments. And fly, spider, you name it. Many of them have this process of elongation with segmentation. So this is what is described here. Okay, so yeah. Okay, and so of course this can give very interesting animals, such as the snake. And so in a beautiful paper in Nature, like a bit less than 10 years ago, the Pourquier group studied the segmentation of the snake. And why do I want to tell you about the snake? It's because clearly, the snake has many somites, many segments, 315. So think about fly. You might have seen that fly has only 14 segments. So the snake has, almost more than 20 times more segments than the fly. And so the point I want to make here is that it's likely that the way you form segments in snakes is different than the way you form segments in fly, and I make this a bit more quantitative in 1 second. But you are almost you have more than one order of magnitude more structure to do, and then you probably need to change something. Okay. And so, yeah, okay, I have nothing. So these are just other examples of embryos, and yeah, so maybe introducing some jargon. So yeah, also you see they're very, very close to each other when they form segments. So this is a mass embryo. So you see the somites that are formed here. The tail bud is here, so the tail bud is growing in this direction, and as it grows, something is happening here, and we'll discuss what is happening here. And then, so this is a region called the presomatic mesoderm. We'll see what happens in the presomatic mesoderm. And then in the anterior to the PSM, presomatic mesoderm, you form segments. Okay, so it's the same in mouse, it's essentially the same in zebrafish, and also in snake. And so here they stained for the future segments. And so you see there is this beautiful pattern of segmentation, which is very much like Eve in fly. Okay? Is it clear for everybody? Okay, so now how would you do that? So in fly, John Reynis introduced you to the French flag model. So if you remember the French flag model, the way it works is that you have a morphogene, so say bicoid, and then bicoid is you define different threshold of bicoids, like, I don't know, some concentration, twice this concentration, and based on these thresholds of concentration, you're going to activate different genes, and these different genes are going to define different regions. And so then you imagine that if you have, several intermingled the French flag model, you could easily do segments, many segments. Yes? Oh, a morphogene. What is a morphogene? Okay, good. Thanks for your question. So a morphogene, it was actually a notion that was introduced by Turing in his first paper where it describes the Turing pattern. A morphogene is essentially a protein or a substance. It doesn't have to be a protein so that when you vary its levels, it's going to order different fates of the cell that are exposed to this substance. So so essentially it's this so what that means is that so for instance, let's think about the example you saw is bicoid hunchback probably. So bicoid is like this as a function of x. Okay, so then what you have is that you can define a threshold of bicoid somewhere so that you activate hunchback. Okay, and so then the fate of the cell that you decide depend on if you have expressed hunchback or not. So that will be, roughly, there will be thoracic versus abdominal. Okay, and so bicoid. So this bicoid is a morphogene in this context because all the cells that are exposed to a concentration of bicoid above this critical concentration are becoming thoracic, or in the head. I mean, of course, there is other thresholds here, where all the cells below this threshold are becoming abdominal. So it's a morphogene because the concentration decides what is the fate of the cell. This is what is a morphogene by definition. Okay? Any other question? Okay, so in the French flag model, so the idea behind the French flag model is that when you have a morphogene, you might have many of those thresholds like that, and these different thresholds will allow you to define different fates. Okay, the good thing with the morphogene idea is that it can, so for instance, I told you about scaling, between the different animals, the hummingbird and the whale. The good thing about the French flag model is that it scales perfectly well. If I take an animal that is twice as small, and even with twice as few cells, so look at, for instance, compare this guy has 4 cells here versus 8 cells here. But then if I change the slope of the morphogene gradient, then you can have the right proportion of every single cell of different fates in the embryo. So that's why it's also an appealing model. It's because it scales easily and can explain why different animals with different size have the same body plan, because you can scale the morphogene. So that's an important property in all these developmental networks. You want them to be robust to perturbation like this, such as scaling. Okay, so now the problem. Why is the morphogene, the French flag model, is probably not the correct model for vertebrates? So in the fly, I've just told you, down the road you're going to define 14 segments. So you can make the calculation or whatever. Like people who have specialized on this are Thomas Gregor and Bill Bialek. They try to quantify how many, what is information content, how many threshold essentially you can define. So if you have 14 segments, it means that you need to define 14 threshold of concentration, say something like that roughly. And these 14 threshold are going to correspond to the 14 segments. So that's okay, but now think about the snake. In the snake, you have 300 segments. So it means that you need to define 300 threshold of concentration in your body. That sounds like a bit too much. Imagine a signal so that you can define 300 different thresholds to define the fate of the cells. That's a bit implausible that you have an animal, that you have a system doing this. And so, the French flag model cannot work for I mean, based on this very, hand-wavy consideration, people thought that the French flag model is not very plausible for, say, the vertebrae in the snake. And so that led Cook and Zeman to propose an interesting model in 1976, which is called the Clock and Wavefront model. So what is the Clock and Wavefront model? So the idea is that instead of having a French flag model where you have a morphogene, the idea is to use the fact that you have growth in the embryo to define some essentially developmental threshold. Essentially, the 300 levels are not the levels of a morphogene. They're going to be 300 steps in development. And that makes more sense because it's easier to envision many steps, different steps in development rather than 300 threshold in a gradient. And so the idea is that they have, as development is going on, you're going to define essentially different checkpoints in development. And you could have many of them. So, I mean, for the segmentation, you could imagine that these checkpoints are going to be linearly distributed, but that they do not have to be linearly in time, but they do not have to be. And so then the idea is that you create a temporal coordinate in development. So you have a time. There is a natural time happening in development. Okay, and so now what happens is that if you have a linear time in development, what you can do is that the idea is to couple this to an actual clock. So what do they mean by a clock? So I'm going so this sketch is going to illustrate what is happening. So you have an embryo. The embryo is developing, okay? And as it develops, what you do is that you have a clock, an oscillator that is oscillating in the posterior part of the embryo. So my blue thing here is a clock. So it's going from low, middle, high concentration, and then the dashed line here represents the linear coordinate in time. So this goes from here to first time, second time, third time. And the idea they have is that when you have this linear coordinate and at the same time the high oscillation of the phase, then you're making a segment. And then once you do the segment, then you go back, and then you have made a segment, and then the linear coordinate is going on. And so if you do that many times, you have a linear coordinate that is progressing along the embryo. So they call that a wavefront. And this wavefront is going to interact with an oscillator. And the interaction between this oscillator and a wavefront is going to create a pattern of vertebrae. So if it's not entirely clear, be reassured, it's normal, because they really present this model without math, too much math, so it's very difficult to get you hold of it. But later on, I will show you an explicit model that will be much clearer, hopefully. So part of the game is actually to clarify these questions. What do we mean by wavefront? What do we mean by, oscillator and stuff like that? But, I wanted to give you also an historical perspective from the biological standpoint, how people think about this question. One of the important things that biologists retain from this clock and wavefront model is that there is a clock. That's an important, and that's something that is quite easy to understand. They assume there is an oscillator. And so people looked for these oscillators in actual embryos, and they found them. And so this is a zebrafish embryo. No, this is, I have a doubt. No, this is both mouse embryos. This is mouse embryos. So this is the tailbud of the mouse that is growing. This is extension of the tailbud, so the embryo growing in both cases. And so what they did is that they reported for some genes, and what you see this beautiful oscillation in the tailbud, and you have this kind of wave. The wave propagate along the presomatic mesoderm and somehow stabilize or do something. We don't quite know what they do at this stage. They do something in the anterior. So this one is a bit more, it's probably a bit better. It came later on. So you have growth of the embryo. And so, and you have a wave, you have a wave propagating. You don't really see the oscillator in the tail because it has very low amplitude, but clearly there is something that is oscillating and that will give you wave of genetic expression. And if you focus there, you might be able to see the boundaries of the vertebrae forming as a wave. So if you look at the wave is going up, And when there is a very high intensity, this is where you move anterior to it, you form the segments. You need to be a bit used to that to see this, but it's pretty clear on this movie. So beautiful movies. And these movies are very recent, like because 10 years ago it was not possible to do such movies. And now people are obviously very interested in all this real-time imaging and looking at developmental process. Okay. So yes, reflected light. No, it's GFP. It's GFP. So actually, it's not GFP because you need to destabilize it because GFP is too stable. So it's destabilized GFP so that you can see the oscillation. So the lifetime is around 2 hours of the GFP protein. So here, what you actually do is that you have a reporter, GFP reporter for a gene called lunatic fringe, which is a gene in the notch signaling pathway. But there are, this is for mouse in zebrafish that did actual fusion protein. So where you really monitor the real oscillations. Yeah, all of this is, these people are biochemical or molecular biology genius. I mean, it took them 5 years to do a line like this, another 5 years to do a line where you have a wind reporter. It's amazing. It's an amount of work that you can. It's very difficult to envision. But now with CRISPR, it's a bit easier to do this stuff. But still, you need to engineer the reporter so that it has the right lifetime and stuff like that. And you will have many things that will not make it work for various reasons. It's very difficult. Anyway. Okay, so obviously I'm telling you about this because we're going to see, going back to our network idea. The big question is that how can you explain this with a network model? Can I use these simple modules that I've told you earlier to model this, and can you evolve a network doing this? That's really the question I'm going to ask. And so, again, to insist on this, so this is a zebrafish now, and so this is a gene called delta, which is also in the notch signaling pathway, that is stained. And here, the good thing with the zebrafish is that you see delta is expressed in both phases of development. It's expressed in the tail and it's going to oscillate. So here it's at a given time, so you don't see the oscillation, but there is the oscillation of delta in the posterior. You have one cycle per 30 minutes at temperature. And then, sorry, no, it's not at room temperature, it's 37 degrees. It's lower at room temperature. And then what happens is that, so you will have the wave of genetic expression of delta going from the tail to the anterior. And then somewhere, something is going to happen, and we don't quite know what. So in the clock and wavefront model, we assume there is a wavefront that is somehow doing this transformation, and basically the clock stops. And the good thing with delta is that you see that you have a pattern, stripe pattern of delta, very much like the segments in the Eve in fly, say. And so the idea is that you have here a kind of temporal oscillation that is transformed into a spatial oscillation, like a whole pattern. And so all the games that how do you do this? How do you do the oscillation first? And how do you make a temporal oscillation that is transformed into a spatial pattern? And the important thing is also you have growth. So it's coupled to growth of the embryo. So that's a complicated thing, but we'll see that it might not be that, maybe a bit less complicated than what it looks like. Okay, so now I got interested into this problem for various reasons, and I thought that I could use this evolutionary computation or evolutionary algorithm to essentially evolve a network performing this process of segmentation. And so to do that, so I describe a bit what I did in terms of mathematical formalism. So what you do, so I'm going to model an embryo as a line of cells. And so in each of these cells, what happens is that I have a gene network, and the gene network is identical in every cell. Okay. And so to account for the growth, I'm going to assume that there is a growth of the embryo. And so to do that, I'm going to impose that there is a region that is growing, like a tail. And to do that very concretely, I'm going to assume that I have, this is what this movie describes actually. So I have my position here in the embryo. X. Okay, and then I'm going to assume that there is a wave like this that is moving along my embryo. So a wave with a line with a speed v, and so that's just a gene that is more concentrated. I impose the shape of a gene to do like that and to sweep across the embryo. So it's like bicoid. It's like moving bicoid. You saw bicoid. It's like I assume that there is a bicoid that is moving along the embryo. Okay, so I'm going to assume that. And then I'm going to give this to my evolutionary algorithm, and I want to evolve a computer that is, to evolve in the computer a network that is able to form stripes of genetic expression. So that's what I'm doing, because eventually I want segments. And what is defining segments, as you saw in the fly examples, are stripes of genetic expression. Okay, and so now I need to, so how do I define that I want more stripe? Well, what I have, so this is my, if you want, this is my input. This is my input. Okay, and so I have, going on there, I have my input. I have network dynamics, and I don't know what it is yet, because this is what I want to evolve. It's kind of a black box. And then I have some output gene. Okay, and so what I'm interested in, I impose the input. I want to evolve the network dynamics, but I put the selective pressure on the output gene. And what I want to do for the output gene, I want to do a pattern. I want to do stripes. So I do not impose anything about what the position of stripe or stuff like that. I just say to my computer, I want stripes of an output gene. And I don't care how you do stripes, you should just give me stripes. That's the idea. And to more precisely, to quantify that, I do something very, very crude. So I'm going to assume that my output gene, so assume you have a profile of the output gene as a function of position that looks like that, at steady state. So what I do, I basically going to count how many transitions there are from between a low concentration and a high concentration. So for instance, if I have a profile like this, I am going to tell there is 1, 2, 3 transition, and so I'm going to score this network. I say yeah, it has scored 3. And the goal of my evolutionary algorithm is to build a network that is going to increase this number of stripes so that at the end of my evolutionary process, I have a network with many, 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 many stripes. So that's what I do. So now what I'm going to show you a typical run of my evolutionary algorithm, and you will see how a network is growing and how you do a network, how you end up with a model of this process. Okay, so I'm going to spend a bit of time on this just to set up the convention. So G is my morphogene that I impose, it's my input. E is my output. So these guys are here right from the beginning. In the beginning, they are not connected at all. So this is a movie that is simulating the behavior of this network when G is so when you have a sliding morphogene that is sliding from left to right. So this is really a kind of, it's very much like a developmental movie. It's very similar to this developmental movie I showed you for an actual embryos, except that this is for my simulated embryo. So, and so this is the behavior of the network. So you have this moving G that is just sweeping here and that is simulating the growth of the embryo. And the first step in my evolutionary computation, the first network that is evolving, early step in evolution, you get this thing here. And so you should immediately recognize E that is self-activating. This is my famous bistable system. Where E can exist at steady state either in a low or high configuration. And this is what you see here at steady state. So once G has disappeared, you are at steady state. And at steady state, E can be at a very high concentration or at a very low concentration. And you make one stripe, one boundary between when you have this region where there's low E expression, zero E expression, and high E expression. So this is this very simple network. It's basically a network that is defining a fate of IE based on, if you look at the initial condition, where is this happening? You know, E is activated only from a low threshold of G, and so it's going to stay activated on the right and to stay inactivated on the left. And so this is the first step to have one boundary. This is a segmentation network with only one boundary. So it's a pretty stupid segmentation network. You know, it's very far from having vertebrae, but at least I have one boundary. Okay, is it clear for everybody? Yes. Sorry? Why is it leading, why is higher where? Sorry? On the right side of the front? It goes up and then it stays uniform here. So the question is that why does it stay uniform? Oh, it's because, okay. It's because it's related to something I commented on yesterday. How do I clearly E? So when in this region, E is both activated by G and E. So how do you account for having two activations at the same time? So so no, that's not that. So activation is not additive here. It's more like a maximum of activation. So there's a maximum activation rate, and once you add this activation rate, you cannot go above it. That's why it's not higher in this region. Okay? No, because it's already on. It's basically it's turned on once and it stays on because it's self-sustained because of this bistability and this positive feedback loop. So you just it's very it's essentially once you get above a threshold you stay on. Okay? Is it clear for everybody? Yes. Yeah, sure. Yeah. Yeah. Why? How do you know that the front is coming here? Here? Well, okay, so I impose the front dynamics. The front dynamics is completely imposed. So the way you should think about it maybe is that these different cells, you could think about them completely independently. It's just a matter of initial condition and a matter of G dynamics. So the cell, and to be more precise, so let's consider what happens in one cell here and one cell here. So on the cell on the left, so G of t is something like this. Okay, and so E is is like turning on and maybe dying off, something like that. So that's E of t. In a cell on the right, what you get is something like this. G is I, and then E, and then dying off. And then E is turning on, and then stays on. Okay? So another way to think about it, that the development, what is development, is just you change the dynamics of the input, and then you look at how the fate depends on the dynamics of the input. And this is what is simulating here. X-axis is time here, sorry. Yeah, it's important to make the distinction because we do time and space at the same time. So t, and this is g of t. Okay? So if you have a growth process, you see, if you assume that this g, so if you have a line of g is moving from left to right, every single cell sees different dynamics of g, and based on the dynamics of g they need to take a decision to activate E or not. Okay? Is it clear for everybody? Okay, good. So that's the first step. So now, what do I mean by first step in evolution? It means that when I run an evolutionary computation, when I run my algorithm, this is an early step in the evolution where after 500 generations, actually less than that, after 10 generations, you have this network that is spontaneously appearing. So already there is an evolution process. This, by stability, it appears from evolution. So already evolution is recovering things we've already seen earlier this week. So now, in evolution, something else is happening later on in evolution, and so you start adding a repressor here. And so one thing I can comment on right now is that now you have G, you have a repressor, and what you see here, like this module here, is our famous incoherent feed-forward loop. Because G is activating, you have like one branch is G R, E, and one branch is G over E. Okay, so we see a feed-forward loop appearing, and the effect of the feed-forward loop on the dynamics is going, so I'm going to show you here. So here's what happens. So everything's happening in the beginning. What you saw is that G activates E, but then just after, slightly later, G is activating R. And so this is what you see here. So now here, G is activating R, and R is still active. But then the effect of this is that you have a transient phase where E was activated before being shut down in the cells that are more posterior. So I show you again the movie, but the thing you should see now is that this is making a nice stripe. This is one stripe. This is essentially a stripe network. So this very simple network creates one stripe of E expression. Okay, so let's go back. Everything is happening in the beginning, so focus on the beginning. Okay, E is up, R is on, but then R was on slightly later, so that you still have this region where E was active, but then R was turning on. R turns on and represses E, and now you have one stripe. Okay? So that's a very simple network. Again, and then you see the feed-forward loop network is appearing very naturally in this evolutionary computation. Again, remember, I'm not forcing anything in terms of network. I just want to select for patterns. And so when I select for patterns, I see this feed-forward loop coupled to a bistable system appearing completely naturally. It's very reproducible. If you go download my software, run the simulation, it will appear first run. It's super, super reproducible. There is really like a very strong evolutionary direction leading to that. Okay, actually there is even a video. I made a YouTube video where I've done this exactly. Okay, so now it's good actually, because we went we wanted to select for pattern. We have one stripe, so that's good. It's a very good start. We have one stripe. So now what is very surprising in this game of evolution in the computer is that the next step, you get essentially the solution in one move, only one move. And the move you're doing is adding a negative feedback loop with delay. So now it's a self-repressing gene that we saw earlier this week. And as I told you, the self-repressing gene with delay, one of the effects is to do an oscillator. So now let's look at the dynamics. So now R is oscillating. R is oscillating, and so you have the front is moving, activating E, and then R is oscillating, and so depending on the value of R when the front is passing, E can stabilize in the up state or in the bottom state. And so that gives you basically, a simple model of stripe formation in vertebrates. Okay, I show it to you again. So you see R is oscillating. R is repressing E, so E is also oscillating, antiphase to R. And then based on what happens when so the important thing is that the oscillators is dying off at some point. So R is dying off, and based on what is the concentration of R when it dies off, you either get the up state or the down state for E, and that make many stripes. So in one evolutionary move, boom, you get many stripes. So that's pretty cool. Yes? Yes, there's a delay. You need to have a delay. We can discuss, I think next slide I have an example without delay where you will see what happens. That makes it more complicated. But you see this is very simple network. It's a 3G networks. It combines three of the modules I've described in the previous lecture. So there is a positive feedback loop, small network that is bistable. And if you see how it appeared in evolution, you need this bistability to define the phase of your cell on off. Upstream on it, there is a feed-forward loop network, GRE. So GRE is the submodule that is defining the stripe. So, and then, once you have a stripe module, upstream of this stripe module, essentially you make a clock with a negative feedback oscillator. And so this is what gives you this very nice and complicated pattern where you have morphogene moving, clock, stabilization of the pattern that make many stripes. So, I told you earlier during the week that even very simple gene networks can have very complicated dynamics. I mean, clearly this is an example. This is a super simple network, but the dynamics are clearly, not trivial and very interesting from a biological standpoint. Okay. So this guy, if you run your evolution simulation, it might not happen all the time, but it happens in a quite reproducible way and for a good fraction of the time. You can have alternative pathway leading to the same kind of solution. So I'm going to start with, so this is another pathway that is evolving. When you run many, yes, sorry.
Sorry. So when does it stop?
Yes. When does it stop what? The clock or the oscillating? It stops when G, so you see you have this morphogene G, and when it drops below a concentration, it no longer activates R, and R stops oscillating. R just dies. Okay? So in the real embryo, this will correspond to the anterior part of the embryo, this will correspond to the posterior part of the embryo. Yes? Yeah, you're right, absolutely. There is no wave here. That's a good observation. There is no wave. What you only see is you only get the clock and the stripes, but you don't have waves. So this is an extremely important biological question. Why are there waves? And this is a question we're currently working on. This is a simplified model. We can refine it to get waves, and there are biological interpretations of why there are waves. But I can tell you later how you could get waves easily with this. It's not difficult. But remember, what I'm doing here, I'm selecting for stripes. So, I get what I select for. I wanted stripe, I get stripes. Now, why would you need waves? One idea that we have is that waves are actually important to pattern the stripes. Because if you look at the stripe here, there is no polarity at all. Up, down, I could make a mirror image of the stripes that would give me exactly the same thing. So, and what we believe in biology, it's not what happens. There is a polarity. There's anterior, posterior polarity. And so this is why you need wave, because the waves are going to give you a directionality. And so this is a simplified vision of segmentation, but the real segmentation is probably more complicated. That said, you can take this model, modify it to also have these waves. It's not difficult to do. Any other question? Yes? No, you don't need spatial diffusion to get that. You can add spatial diffusion. You can make the evolutionary simulation with spatial diffusion. You will still get the same result. Yeah, but okay. Remember, the game here, I'm taking a rather reductionist approach in the sense that the game here is that I give a phenotype, which is making stripes. I want to see what kind of networks can give my phenotypes, and what do I get from these networks. So for instance, one thing you can notice from this, I never selected for a clock. I never said I should have a clock. But the clock appeared very spontaneously in evolution. So that means that if I want to have stripes coupled to growth, say, the clock solution is very natural and you expect it to evolve. Now, if I want to add constraints such as I want polarity within stripe or stuff like that, you might have other things evolving. But I would say, it's becoming a first-order effect. I'm looking at the zero-order effect, which is having these very simple networks, and then at first order you can, and we've been, I probably won't have time to talk about fly mosquito later on, but a little bit. But this is an equation we also add in this context. So, okay. So the size of the stripes here, it's a very simple thing. It's pure dimensional analysis. So what is the size of the stripe? I can do it by dimensional analysis. So there is a speed v, which is the speed of propagation of the front. And then the clock, what it gives you a time scale t. Okay? So how do you make a size with a speed and a time? You just multiply. The size of the stripes is actually speed times period of the oscillator. So if I increase the period of the oscillator, I will have a bigger segments. If I decrease the period of the oscillator, I would get a fast oscillating system, and you have smaller segments. Yeah. This size here is the distance between. Okay, I don't have enough precision to do it on the slides, but you have your stripes like this. This L here is the distance between the left side of one stripe and the left side of the next stripe. What regulates the relative size of up and down things, this is what happens right at the bifurcation so you need to look at the detail of the interaction of the oscillator with the bistable system. You need to be a bit more precise. Yes? It's very difficult to have. Yeah. Yeah. Okay. Actually, so the speed of the front. That's a very good question too. Okay, so it's a kind of a Pandora box. At short time scale, a few segments, the segments have the same size. You know, like if you look at in the movies I showed you, when you see you see that the segment has the same size, which means that the front here is basically propagating at roughly the same speed. Now, during the development itself, you are going to make segments of different size. So that means that indeed the speed is going to change. But basically, the speed is changing at a much lower time scale than the clock period. So which means that this guy here is an approximation. At short time scale, this is going to work like this. But then you can modulate the speed of the front later on and get segments of different size. That said, the question you ask is an extremely important question, and you need to revise this a little bit because, for instance, it might be that when you change the speed of the front, this very neat process of having segments, turning on, off. Maybe if you change the speed of the gradient, it's no longer going to work because this network that evolved was actually tuned to react to this speed of the gradient. So you might have a different mechanism that needs to be robust against this variation of the speed of the gradient. We are working on that right now. I have a solution. It's actually what your question makes me think I should evolve this in the computer explicitly to see if the solution I have is the one that is evolving the computer. But we're thinking about that. The last thing, I told you open a Pandora box, so I'm going to really discuss everything. There is absolutely no evidence yet that the speed of the front is connected to the growth. Actually, we have a paper where we show it's not true. We can stop the growth and we still see a front propagating, but the speed of the front is becoming exponentially slow. So there is a weird connection between growth and speed of the front. Again, that's something we're working on right now, and I think we understand it now, but in the simple model, this is not happening. Yes? Yeah, I have imposed it, but in the real system, but then, this is, again, it's a reductionist approach. I start with a simple assumption, see what I get, and then we can add hypotheses to fit more and more biology. The goal is to explain biology, of course, obviously. So anyway. Other questions on that? Okay. So I told you, so when you do these evolutionary games, you have all the things happening that can happen. So for instance, this is another alternative pathway. And so this is the second step in what I showed you, the module that is making one stripe, feed forward plus bistable equals stripe. And then what you do here, so what happens is this evolutionary simulation, in this specific evolutionary simulation, is that instead of making an oscillator with a mutual repression, with a self-repression with delay, the network, the evolutionary algorithm added another stripe, another repressor here upstream of the first repressor. So start having a cascade of repressor. So then the effect of this, you can see it here, is that now if this R2 is repressed a bit after R1, you basically turn on again E, and so you make a system with essentially now one stripe plus one domain. Okay. And then once you have a cascade of repressors, actually it's quite easy to get to get an oscillator, you just actually quite easy, no, because you need to add another repressor that is activated by E, and then what you have here, you get an oscillator which is a repressilator I discussed yesterday, which is very similar to the repressilator that was done by Elovitz and Neibler. So the bottom line of this is that there are several ways. There are several ways to the same result. You still get an oscillator in the end, but the network associated to this oscillator is different. And I think that's a general lesson. When you select for phenotype, you might get you might get different networks, but the networks are going to have very, very similar phenotypes. And one of the questions is how you relate this phenotype and these networks to each other. Clearly, this network is different from that network. It's obvious it's not the same network. Here I have one repressor, but part of these networks, you still have commonalities between those networks. So how do you map one to the other? I think it's an interesting question. And like the early stage of evolution, where you add this bistable and then this R1 repressing this bistable system, are the same. So you also have commonalities. So this is why you do this simulation. It's just to somehow see and quantify a bit, get intuition on how evolution can work to select a precise phenotype. Yep. Yeah. Yeah. Yeah, it's because you also adjust parameters. So you could have some kind of a bit of hitchhiking too, like you lose the first stripe, but since you gain other stripe in the posterior, that doesn't matter. The important thing also, I told you, I select on the number of stripes, the number of patterns. So I don't care where they are. So it doesn't matter if I lose one stripe here and then get one stripe in the back, it's the same score. So that's actually very important in these simulations to get fitness functions that are kind of robust to this kind of changes. Because if you were to say, I want a stripe here, that will constrain too much evolution and you will not be able to find a network optimized for this. So it's better to do that this way, to select on general features of the network to get results. And then, of course, you can say, you can change parameters and see what you get. Good. Don't remember. Okay, so yeah, so that's a slide about stochasticity versus determinism in this evolutionary simulation. So as I told you, something are variable from one simulation to the other, something are the same. So what is stochastic? What is variable is the network topology. It's not exactly the same. So, you can have different ways to get an oscillator, you can have different ways to get a bistable system. This also happens from time to time. What is reproducible from one simulation to the other? It's more like the geometry, the dynamics of what is happening, which is sketched here. So what is this graph? What does this graph mean? So it's basically summarizes the fact that in all the simulation you have an oscillator coupled to a bistable system. And you don't really care how this coupling is done and how the bistable system or how the oscillating system is done. What matters is that they are coupled in a way so that so FGF will be like my morphogene, sorry about that. If you are at a high morphogene concentration, you want to have an oscillator, okay? And then if you want to have at low morphogene concentration, you want to have a bistable system. Then something happened in between, but certainly like the endpoints of this evolution, oscillator and bistable, we see them in all simulations. We always see, or at least the simulations that are successful, that gives you many stripes. You always see an oscillating system coupled to a bistable system. What happens in between, it's not entirely clear. The standard thing would be to have a half bifurcation and then, a saddle node or a pitchfork bifurcation. And actually, we're studying right now what is happening if you change a bit this picture, how you modulate what is inside and how that would change your result. But the endpoints are very clear, and this is what you see in all simulations. And the point I want to make there is that this is the right level to study these problems. I mean, I strongly believe this is the right level to study this problem. You want to study this problem at the level of essentially the bifurcation diagrams. Sorry. Okay, which is exactly what I've just told. Okay, so I'm going to summarize this. So all of this was published in a paper 10 years ago. So summary, what do we learn from this simulation? So first is that we wanted to simulate a pattern formation in a growing embryo similar to vertebrate, and we get results which are quite reproducible from one simulation to the other. What we see is that when we run many simulations, you basically get convergent evolution on the same network organization. So the way it works is that not only you get convergent evolution to the clock and bistable thing, but the steps of evolution are also the same. So you first start to make a bistable system, then you make a repressor to repress the bistable system, make one stripe. Then once you have one stripe, you make an oscillator to basically multiply the stripe. So you really get not only this is parallel convergent evolution to the same solution, but it's a really parallel evolution. So the steps are also the same when you reproduce this evolution, which makes our results robust, at least in this context. We think that this is in this context with the hypothesis we give, this is a robust solution that you will find, and maybe this is what happened also in real evolution. So we get many solutions. This phenotype is similar, so you always get this clock coupled to the bistable system. So a very important and subtle thing that I've not described very explicitly, but now I'm doing it now, is that we get this clock and bistable system, but I did not select for a clock. We know there is a clock in the real system, but the clock is just appearing very naturally from this evolutionary simulation. So, and this realizes basically something very similar. It's basically a clock on wavefront model. So I told you with this little sketches, the clock on wavefront thing, I told you it might be difficult to understand. I hope you understand it better now, because with the gene network approach, simple things, you get a clock on wavefront model essentially for free. So we selected for pattern formation in a growing embryo, boom, the solution you get is a clock on wavefront model. I think it's quite nice. And it tells you something, about real biology. This is something that is very systematic in all evolution simulation that we have been doing. Whenever we select for a complicated phenotype, very often we get results where some things that you actually observed in biology that you have not selected for appear for free. So for instance, the clock appears for free. We just get it. We did not select for it, but we get it for free. And I give you another example in my second lecture on immunology. And so at this time, it was the first model of the complete system, because people were mostly focused on the clock, but they did not really were not that interested in what happens and how the clock is read by the system. But 10 years ago, it was one of the first models. Yes? Yeah. Yeah, this is this drawing here. I mean, when I drew this, I'm selecting for the pattern of the output gene, and I'm selecting I want as many stripes as possible. Yeah, just the final pattern. I'm not saying you cannot get it from Turing pattern. I'm just saying it does not evolve in the simulation. I just say the clock on wavefront model is easier to evolve in the simulations. That's just what I say. It's not an impossibility theorem about the fact of having a Turing morphogenesis. I'm just telling you, if I do these simulations and I want to select for a pattern, it's super easy to make a clock on wavefront model. Yeah. But the thing that why would I select for space-time? If I select for space, I already get what I want. I don't want to constrain. I think, in general, in the simulations, you should not constrain. What is very interesting is that you don't constrain it. You just say something very minimal, and then you see what you get. And then if you get more refined stuff, you can get more constraints. But I think it's very interesting to see what happens when you don't constrain it precisely. I'm not claiming that nothing else is possible. I'm just claiming that if I make, I'm a physicist. Minimal things, what is the barest minimal thing I can get with a minimum number of hypotheses? What can I evolve? I get this. This is a statement I make. And it turns out that it's not that far from the real system, which is interesting, I think. Yes? Yeah. 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 No. No, I'm very stupid. Everything I will tell you today, I've not figured them out before.
So you actually found out the simulation?
Yeah. Yeah, yeah. So this network, it appeared in the evolutionary simulation. I did not think about it. I actually, but okay. So the full story is that I had thought that a clock coupled to a bistable system might be the solution, and so I tried to build a model by myself. And it was horribly complicated. I know I could not get something simple. This model is much simpler than anything that I've done. Yeah, yeah. Could they be biased in any way? So the bias, I think the strong bias you have, the main bias you have is a biochemical grammar that you allow yourself to have. So in the sense that, I say I can have basically here it's the transcriptional network, but I'm modeling this with Hill functions, which is not very, it's not very biased hypothesis, but still it's a bias. Because you can have very strong nonlinearity, say. If I were to evolve this with only very linear things, that might not work. Yeah. So the delay certainly helps. So this is much easier to evolve this when you put delays to get the clock. But I've done it too without delays. You don't get the clock because this guy is impossible to oscillate if you don't have delay, but you get a clock that is appearing upstream in a different way. It's just less probable, it just takes more time actually, more evolutionary time to appear, but the geometry of the solution is the same. It's like this bistable, this clock, and appearing in a very, very similar and independent way. So the immune system is even more spectacular from that standpoint, because the solution I got I was simply Flagger blasted by the solution. It was so much simpler than what I had done by hand. I really understood this. In retrospect, I was like, oh, okay, I could have guessed it. But the other one, I could have guessed it, yes, but I was really too stupid to guess it. I just did not see the light first. Like when I hear, I kind of see the geometry, but for the immune system, I did not see it. Okay, yes? Yeah. I don't, okay, no period. No period. I'm just selecting for, I have my fitness. Okay, I'm just counting how many transitions you have from a low to a high region of E. So I want to have regions. So the idea is that I have a line of cells. Some cells are turning, where there's a gene that is on in this cell, a gene is off in this cell. And what I want to have as many as possible alternation between regions of I and a low region of IE and low E. Because it doesn't have to be, I didn't want to impose that I have a pattern. I didn't want to impose that, like this for my evolution simulation. So let's assume, let's look at a network that would have a four pattern, or fitness 4. So this would be a network, pattern where I have fitness 4, so that's relatively regular. But this guy, these two guys in my simulation, they have the exact same score. They are no different at all, because they have 1, 2, 3, 4, 1, 2, 3, 4 transitions. I'm not selecting on position or size of the pattern. And actually, as you feel, it's really remarkable that we get a rather periodic pattern, because this is not something that we impose at all. It just came for free, and it comes from free because of the clock. Okay. Okay. I have 10 minutes. Okay. So. In the last 10 minutes, I don't have time to tell you about the fly mosquito thing, unfortunately. So I'm going to tell you, it's a bit the same story, but for Hox gene patterning. So Hox gene patterning, so this is I don't know if you've heard about it. Hox gene patterning, this is, if you have different animals, you have the different regions are defined by different Hox genes. So, so for instance, these genes here, and obviously it's coupled to segmentation, but you have specific genes that are expressed in the thorax, in the abdomen, in the head, that are defining different regions of the embryo. And so essentially the gap genes are derived from Hox genes. So it's very similar to gap genes. So the way you should think about it is the following. So it's a bit like segments, except these are segments with different genes, basically. So Hox genes, they are expressed at different positions, and some you have some specific Hox genes expressed at any given position. And so our goal, we try to see what kind of network could do that. What kind of networks could do this kind of pattern. And so I'm going to skip the math because I don't have time. I'm just going to show you the results. So now the simulations is this. Yeah, you still have my morpho gene that is propagating. So it's like, again, my growth zone. And what I want basically cells of different colors. So I have many, many different genes, and I want that in a given cell, only one gene is expressed. So that's what I do. That's roughly what I do. It's not exactly what I do, but I don't have time to go into the details of that. I want to show you the results. That's why I don't want to go too much into the details of how we do that, the fitness and all of this. So this is another simulation. This is the final result of my network, of my evolutionary simulation. So this is a network that is evolving. And so the first observation you can have is that it's a very complicated network. But even though it's complicated, you see there is some kind of structure. There is some kind of hierarchy in these networks. Okay? The second thing you should notice is that, so the color here of the genes are the same as the color here. So the very bottom gene in the network is the first to be expressed. The second to be expressed here is here, so it's 4 is here. Now 5 is just upstream these two guys, and it's here. And then you see that there is essentially, as you progress in time, you also progress in the direction of the network topology. And so, of course, the network are doing what you expect them to do, which is in any given position, you just express one of these genes. And so you see the very last one, and now here, I don't know if there's 5 or 6. So this guy here is just the one that is upstream everybody else. Okay? So this is clearly not a random network. It's structured, and the way the network is structured corresponds to the way the order of genes that are expressed. Again, this evolved in the computer. I'm not cheating, and I'm not cheating. It's really something that came naturally from the evolutionary simulation. You always get this bistable thing. The bistable things are just here to fix the fate. So it's not complete. Now you should be familiar with this. When you have a bistable system, you just say it's one fate. And then what happens is that what you see from this network is that, for instance, the gene 5 here, it's repressing the gene 4 and the gene 2. So gene 5 here, it represses the gene. So essentially, the genes on the right repress the gene on the left. So the gene 5 is repressing gene 2, gene 4. Now, if you look at gene, so maybe gene, so it's not always direct. So for instance, this gene 8, which is here, is repressing gene 5. And then you have something upstream of 8 that is repressing the gene 2. So it's not always exactly, there's not a simple rule, but you can quantify this, and on average, you will see that the genes on the left are repressed by the gene on the right, and the more on the right you are, the less you are repressed. So you get rules like this that are evolving very spontaneously. And the reason why I mention this is because this rule corresponds to an actual rule in developmental biology, which is something called the posterior prevalence. So these are you might have seen these mutant flies, where you basically transform an Anterior. So these Anterior are the things that are helping the fly to balance itself. So you make a Hox gene mutation, you mutate one Hox gene, and suddenly in the adult fly, you get another pair that is appearing. So this is something called posterior prevalence because this is represented here. So imagine 8 is the gene corresponding to Anterior, and 5 is the gene corresponding to wings. So now if I kill 8, imagine I kill 8, since 8 is replacing 5, 5 is going to extend here. And so now you have a region where you should have wings, which is essentially doubled. And so this is why you transform this Anterior region into this Eve thing. into these wing things. So again, the point here is that evolutionary computation is able to recover. I mean, this is clearly a high-level phenotype. I mean, if I had told you, consider a fly, would I expect this? You'd have, well, it looks kind of random. Why would you have this? This posterior prevalence rule, people know it. This is something that has been observed, and it's more like, and it's not completely exact too, but this is something that people know, and there is no clear explanation for it. It's just a rule. But now what you see that when you do this evolutionary simulation, given some hypothesis, and I do not have time to go too much into the detail of the hypothesis, but you see these rules also appear from very simple evolutionary simulations. So this is how this is, I think it's quite interesting, again, that you learn this kind of stuff by performing the simulation, that you can reproduce this kind of high-level rules. So we can make this more quantitative. I should say that we understand the posterior prevalence rule, and the reason why essentially the reason is that these are the networks, the network topology that we expect are kind of always the same idea of bistability coupled to repression, making feed-forward loop. And you can show, so for example, you can take this network here, and you can randomly change parameters, and you can show that the more probable pattern that you would expect is that gene 1 is going to be the most right one, gene 2 here, the middle one, gene 3, the more anterior one. You can show that, if you take this network, change parameter, which is typically what you would expect to evolve, what you see to evolve, it will be associated to a pattern. There is not a 100% correspondence. These things mean, this is what happens for the location of this tribe when you randomize parameters, and you see there are some differences. But we can rationalize a posteriori why this is what we observe. But again, we learned that from the evolutionary computations. It's not something we put by hand. We see it emerging from our simulations. Okay. I don't have time to tell you about all of this, but essentially what is really interesting is, again, some kind of meta rules that you see in development, such as posterior dominance. We can basically understand them by evolutionary computation on top of network topologies and stuff like that. So that was published 7 years ago. With this, I think I'm going to wrap up for now, and I see you in 30 minutes. So if there is any question? Yes, okay, there are questions, yes. Yeah.
We have the starting like a G and E concentration.
Yeah.
Do we have biological analogs in the biological systems so that we know that
so
this
gene and E, yeah. So like this gene G and E, yeah, you have analog. So G is a morphogene. There are many known morphogenes in the posterior that are sweeping across the embryo, like FGF, caudal, wind. There's no lack of candidate. For the bistable thing, it's the same. We have, there's notch delta signaling that is clearly implicated in the definition of the stripes. So notch delta will be a good candidate for the bistability. The real problem we have actually that there are too many candidates. So it's likely, like if you, and it's a huge problem in this field because everything is oscillating, everything looks bistable, and clearly also if you make a mutant for one gene in one species, it does not do anything like essentially the networks are probably different between species. So that's also why it's important to do this kind of work, because it's not, the networks are not conserved. The networks themselves are not conserved. It's more that the function of the networks that is conserved, and so this is why I think you need to have a functional understanding above all, rather than a specific network topology understanding. That's the other reason why I'm not too much on, network in general. Like networks are probably specific. The mouse network is not the human network. I'm pretty sure about that.
And it is like super interesting to have posterior dominance coming out of nowhere.
Yeah.
So does it tell us about the actual evolutionary process that?
I don't know. So this kind of simulations, there are two ways you can think about it. You can think about it as simulation of actual evolutions, and biologists are killing me when I say that, because they say evolution is more complicated. Or you could think about it as more like, computer-aided design of actual gene networks, where you recover some properties you see what kind of, given some rules, what kind of networks you expect to get. So I think the second one is somehow slightly more reasonable. And so what it tells you is that if I give you this kind of dynamics for the input and this kind of rules for the network, then it's more probable to have posterior dominance because it's just more robust to parameter variations and stuff like that. So you can see it this way, and then you cannot make any statement about evolution. I guess this is an evolution conference, so I should not say that, but you know, it's still informative, I think.
Yeah, so you first said that like the goal is to always go back to the biology of it.
Yeah,
right?
yeah.
So in this case, it's not clear that what are we thinking in terms of biology when
okay, so going back to biology, because I was coming next, but I won't do it because I want to focus on but we make some very specific, you know, you see from the slides, we have been studying also actual biological problems. And so one thing we did in this, I mean, I have one slide of prediction versus, so we basically predict, so we looked at the pattern. So in a nutshell, what we've done is that we look at the fly segmentation network, and we looked at mosquito that has a different phenotype, and we try to infer the mosquito segmentation network first. And then once we infer the mosquito segmentation network, we try to see what is the intermediate species and try to predict an intermediate species between fly and mosquito. So it's very much like going back in time. It's like you run back the evolutionary tape to the ancestor of both fly and mosquito, and then you move forward to predict another species. And so, and basically when we do this, we can predict this clogmia, which is an intermediate insect. And so the difference here is that so you compare the relative location of stripes versus a gap gene that you've seen maybe knurps. And so what we see, so we predict the pattern, the correct pattern of clogmia compared to fly. We predict that it's different, and we predict that it's like this, where knurps is basically between 4 and 5, while in fly, knurps is right on top on 5. So that sounds like very detailed, like narrow prediction, but it's actually not. It comes very naturally from the network structure, and this is the kind of prediction we're able to do. Good? Yes.
So the way you do the evolutionary simulations is basically how do you start off with the network? You give it a network structure or?
Okay, so we do both. That's a good question. So for the segmentation, for the somite thing, the clock on wavefront thing, I give nothing. I just gave input, output, and then I let the network evolve everything in between. For these fly mosquito things, I really played the evolutionary tape back. So I gave a fly network, full fly network, and then I went back in time and then moved forward in time. So I gave this network.
So you don't have to define the number of nodes you can have.
No. So again, it was in the spirit of being as minimal as possible. So I gave the fly network and I say just change parameters and see what is happening.
Okay. So let's say iterated process. So first you do select only for the fitness with the number of segments.
Yeah.
And then you have to refine it further to get the proper parameters.
So the fly mosquito thing, which I have not detailed, we play on parameters and we want to refine something. But for the segmentation, like somitogenesis thing, I'm not doing any refinement. Again, I just want to have a pattern. I don't care the shape of the pattern, the position of the stripes, all of that.
Yeah, that gives you the network structure which is observed and you get all
the
information which you finally show us is more uniform. So
no. It's straight from the evolutionary computation. The uniformity is purely coming from the fact that there is a clock upstream. I have not refined anything.
You also get those steps, right?
No, these things I get them as intermediate steps, say. But the thing that if you want to, the reason why you don't get stuff like that in the end is because you have imagine your feed of cells is like this. Okay, so now how do you increase the size, or how do you increase the number of patterns? You clearly need to get rid of this big stuff to be able to put more stripes. And that's also why you don't get this, because then you have a clock which is a very, very rapid period that helps you getting rid of these big things. So that's why you don't get that. It's because it's just, it has, in a limited region, it would have pretty bad fitness. While this guy, essentially, another way to say it is that if you have already regular stripes, it has more developmental evolutionary potential to add more stripes than these guys. Okay.